Again, I want to thank also thank the people who organized this to inviting me to come and give a talk. This might come from a slightly different perspective because we are Mosaic Biosciences, and Mosaic Biosciences is more of a device. We'd like to think of ourselves as a device company and not a drug company. However, I think hopefully as you will see at the end of this, we have a device that can be a very useful tool for hopefully delivering drugs and specifically in peptides and using peptides. So Mosaic Biosciences was started in 2009 with the idea of licensing polymer chemistry technology from the University of Colorado at Boulder, from the Department of Chemical and Biological Engineering out of the lab of Christy Anseth, who does tissue engineering, with the idea of using this technology to develop innovative biomaterials for unmet clinical or medical needs in the fields of tissue regeneration and drug delivery. Currently, we are developing and pursuing products in the tissue in regeneration and repair field, adjuncts to tissue cell and stem cell transplants, and also in drug delivery. In the tissue regeneration repair, we basically can make polymers, and this will become more clear as I go on, that can stimulate and scaffold natural tissue healing processes with the idea that you add this material to a certain wound site or a defect, that you can get cells to move into it and develop new tissue in its place. And then the material itself clears and disappears as this regeneration process occurs. A similar issue occurs with tissue and cell transplant. Of course, we can, because of the very mild nature of this chemistry, we can do all of this chemistry, this polymerizations, in the presence of cells and tissues without any damage to them. In which case, we have a delivery system for taking stem cells and putting them into a site of interest, polymerizing around those cells in that defect site, and then allowing the cells to do what they're going to do as far as the stem cells and mediating tissue regeneration. And then also cells can move in from the outside, from the surrounding areas, and degrade the tissue based on how we build the networks. Additionally, we have begun some work with drug delivery companies, and you'll see this later on about how we can tune our materials to specifically and controllably release drugs and deliver drugs under different conditions. And just a point here, we are located in Boulder. And that is one of the reasons why this is such an inviting talk for us, because it's so close. And I hope those of you who are from out of town are enjoying it. So could you start the video, please? I don't have access. It's really impossible to explain our polymer technology unless you get a chance to see it. So what we have is a liquid polymer that we're applying in place, and it's a little bit faded out here, but I think you'll get the idea. Is that we put something on as a liquid, and this is to simulate, let's say, a skin wound, for example, that you would put on the surface, and then we have, within 15 seconds, 15, 20 seconds, we can use a 385 nanometer light at 1 milliwatt per centimeter squared, which is very, very little light. And we can polymerize this thing just like that. And it then conforms to the wound, it conforms to the defect area. You see here two different molds. And you can see the sort of the flexibility of the material. And then here, this last little bit is showing you how we can change the material properties. You can see the one on the right is a little bit more stiff than the one on the left. We can make these things as stiff as bone or glass and as soft as jello. So we have an incredible ability to tune these materials for whatever application we're doing. This polymer chemistry is based on what we call thiolene chemistry. It's, that came out of the University of Colorado, Boulder. The idea here is that you have a thiol group, which everyone should recognize in this group, and then an ene group. Under specific radical conditions, we usually use light. In this case, we use a light photoinitiator that creates a radical at 385 nanometer light. That's the specific one we use. Of course, you could tune this to go out into the UV, lower in the UV spectrum, or up into visible light, doesn't matter. But when you initiate that, you get this thiol group will react with the ene, and you get a covalent linkage between these two. Now, how does this work into polymerizing things in polymers? Well, if you take in this example, we've got a backbone. Let's say this is a 4-arm PEG molecule that we functionalized on each end with one of these reactable ene groups. And we have a dithiol crosslinker. If we mix those two things together in a stoichiometric ratio, and then put the initiator in there with light, we can create a network out of this. The great thing about this is that it's tremendously modular, in that if we build a thiol toolbox and an ene toolbox, we can have thiols that have different shapes and sizes and different numbers of functionalities on them, and the same thing can be done with enes. And with those, once we combine these, as long as we keep the stoichiometric ratio, we can build networks of different sizes and cross-linking. So here you see an example of a 5-arm dithiol K and a 10K 4-arm. You get a network that's approximately 10 by 10. We can also, by using a slightly larger PEG backbone, we can get a 15 by 15. And then if we use these two same ones but add in an additional 5K linear diene, we can then create a much larger network. So you can see here that based on just what ones we pick out of this toolbox, we can create networks however we want. Now, how does this deal with, or how does this, what does this mean to peptide chemistry? Well, everybody here in this group should know what a cysteine is. It has a thiol group. Well, because our chemistry uses thiols, cysteine becomes a very useful tool for us. For example, if we take a linear peptide or polypeptide and we put 2 cysteines in it, we instantly have our dithiol crosslinker. This particular one has a sequence in the middle of it that is degradable by MMP enzymes that are secreted by cells during remodeling. We can also have a tethered moiety. In this case, we put RGD, which is a cell adhesion moiety that cells integrins attached to. We put 1 cysteine on there, we react it with this chemistry, and suddenly we've got a network that cells can hold on to. As you see here, the cells can then, because we've got it all cross-linked with this MMP degradable thing, cells secrete the MMPs and they can break down this network. Additionally, and this is where potentially this group might be more interested in, is that if we put some type of a cargo, and this could be a small molecule drug, it could be another peptide, it could be anything, a growth factor, we tether it with 1 cysteine on one end and we put that MMP degradable peptide in there. What we can end up doing is we can tether this drug into our network. These MMPs that are secreted will be able to cleave that off, and it will deliver it then to the cells. And it will do it in a very localized manner because these MMPs don't get that far away from the cell before they can have this activity. Another great thing about this material is if you're going to be implanting this or putting it in this body is that if in this example we have these different networks, if we have an MMP dithiol crosslinker, when the body breaks this thing down, it will break it down into very small, well-defined products. And these are all, in this case, 20K 4-arm is the biggest one here. This is all cleared renally. So you don't have any big problems of these big PEG molecules or any type of molecules that end up in the liver because they can't get cleared. Additionally, we can tune these networks to degrade by whatever mechanism we want. In this particular example, we can put a dithiol MMP degradable, or 2 cysteines flanking an MMP degradable sequence. We can have a plasma degradable sequence in there. We could also put 2 thiols on the end and have it filled with hydrolytically degradable moieties, in which case it will just break down by water slowly. The nice thing about using these enzymatically degradable moieties is that you know that the cell is going to degrade it when the cell's in the presence or in the area. Hydrolytically degradable things are more of sort of it's just a time and then it's going to fall apart. You can also mix and match these things, so you can put them all together and you can have a network that will degrade by all of these mechanisms. And MMP and plasma, it doesn't matter. You can pick any human neutrophil elastase, whatever you want, any particular peptide sequence based on the enzyme. As for drug delivery approaches, we can tune drug delivery and the release of drugs by a couple of different basic mechanisms. The first being we can change the network size. So if we were to encapsulate the molecules, essentially forming the polymer around the drug of interest, just by altering the network size, we can slow or speed up the rate of release of the molecule. If you get, in this case, if you get a network that's formed and the mesh size is big enough so that the thing will easily diffuse, it will come out fairly quickly. However, if you have a mesh size that's very small and is able to completely entrap the molecule, then what you can do is that only when this thing degrades, and of course you can tune how it degrades, only when it degrades will the drug be released. Another interesting way that we can do this, and I alluded to this earlier, is the covalent attachment. So if we were to tether with a peptide, our drug of interest, incorporate it into the network upon polymerization, this will get released upon cleavage of this moiety. And then the release rate will depend some on the same mechanisms here, mesh size, polymer degradation, but also the rate of cleavage of this particular linkage. Couple of quick examples, and I know I'm out of time, so this is some work that I did while I was still working in Christy Anseth's lab, one of the founding scientists. I used BMP2, I put it into these rat calvaria defects, and what I was able to find when I compare collagen sponge as a delivery vehicle for our hydrogels, I get much greater bone or biological activity out of the BMP2, meaning that this chemistry is good for these for biological proteins and retains their activity. It's also much better. I got these rats just grew these gigantic soccer balls in their heads at the same concentrations that were the therapeutic range for collagen sponge. Another paper that came out that I'm on this is an idea of this tethering, and here you see dexamethasone has been released. We've attached the dexamethasone to a peptide linkage, one of these MMP degradable linkages. And when MMP comes, it cleaves off this end here, cleaves it right here, and because this cysteine is here, you can incorporate it really easily into the network. And this is the end. Just a quick summary, Mosaic is a device company. We have an unbelievably versatile polymer platform. It is great with peptides. In fact, we use peptides in there, and we can tune things to degrade and release active molecules. We can do in situ polymerization into patients. It's a tremendously versatile platform. And to sort of answer Ralph's question from the previous speaker, if there's anyone here who thinks that our technology would be useful for whatever drug that they have or whatever things that they're doing, we would love to talk to you, because we really think that this is going to sort of change the landscape for drug delivery of biologics. And I have a poster tomorrow afternoon, and feel free I'll have all this information up there, and feel free to come by and talk to me. I'd love to hear what you guys have to say about it. With that, I'll take questions.
Thank you very much, Peter. Questions for Mosaic?